Let's go ahead and get started. So there's only 7 questions, so this one's nice and short. The use or modification of living things to improve our lives is called, so is it heredity, biotechnology, genetics, or DNA? Yeah, that was hard. The genetics and heredity, those ones are, so I guess heredity is basically just the ability to pass on the different genetics, or I guess your characteristics and traits. And genetics is just the studying of the genes and kind of how like your genes really, I guess, affect our appearance and our physical traits. Yeah, so biotechnology, think of that as like a biology and technology kind of coming together. So we are modifying our biology, our genes through the use of technology. And it's not necessarily just through like computer sort of stuff, but it's definitely through some scientific technology and equipment that we can use. All right, we're off to a great start, guys. So which of the following is not a method of biotechnology? So carcinogenics, artificial reproductive technology, selective breeding, or genetic engineering. Good stuff. Okay, so the one that is not a method of biotechnology. So the carcinogenics, I actually just came up with that one on my own. In the reading, you guys should have learned about carcinogens, and those are basically toxins or chemicals that can cause cancer. They are one of the ways that DNA can actually be accidentally altered or damaged, but they're not an actual method of biotechnology. So biotechnology is purposeful, so that'd be through selective breeding, where they're breeding, they're choosing 2 parents and bringing them for a specific purpose for a certain trait. There is the genetic engineering, which they're using, they're modifying the egg and sperm cell, and the artificial reproductive technology is the joining of the egg and sperm, typically before before it putting it back into the female or the parent. So soaring dog, you're soaring up to first place. Blank is where a sperm and egg are fertilized in a petri dish, then implanted in a female. So is it selective breeding, in vitro fertilization, genetic engineering, or artificial insemination? All right, good stuff. So it is in vitro fertilization, the artificial insemination. That is where the sperm is actually inserted in, directly into the female. So that's how the two kind of differ, is that the in vitro is grown into petri, is fertilized in the petri dish, whereas the artificial insemination is just implanted directly into the female. Ooh, there we go. Getting a nice little mixture of points. So which of the following is not a risk of biotechnology? So an increased risk in deadly potential disease, crossbreeding can occur and the offspring may be immune or susceptible, or an increase in population of a species. All right, good stuff, guys. Good job. All right, Nimble Ferret still in the lead. Who is Dolly? So is Dolly a hybrid species, a country singer, a cow who was genetically modified, or a sheep who was cloned?
I remember when this happened.
Yeah. I remember this one.
Good. Well, and actually, so from our first assignment that we had in our biology unit before the school closed down, I'd actually brought up the fact that there was this Dolly the sheep that was cloned, although you guys weren't told much about it. But anyway, so yeah, hopefully you guys watched the video about Dolly the sheep. So Dolly the sheep, I think, was made in 1996. And was cloned from an actual sheep. It was kind of sad though, like if you watch the whole video, they talked about how they had they'd gone through like 400 different specimens where they were born and then they just died at birth. So Dolly was really the only one that was successfully cloned and born and lived she didn't live a full life. I think she ended up dying of cancer, I believe it was. So she didn't live a full life, but I think she lived up to be 6 or 7 years old, if memory serves me correct. But anyways, the funny one there about the country singers is that's actually where Dolly got her name from, was from Dolly Parton. So the Scottish scientist named her after the country singer. So
That's awesome.
all right, good stuff here. Good stuff. What organism was cloned before Dolly? So this one here was also part of that video. So was it a frog, a lizard, a cat, or a spider?
No, it was not you, Gus.
All right, so so it was a frog, and the frog I think was cloned in the 60s, and they just never, for the most part, they never really attempted to clone other organisms, or I guess mammals and animals, just because they thought it was too complex. And then finally, Dolly worked out. But yeah, it was cool. So the frog was cloned in the 60s. I was kind of interested when they said that was cloned in the 60s. I didn't think that technology could be so advanced, or that they would even have that much of an understanding of cloning at that time. So I thought that was really cool. All right, Nimble Ferret, we're doing great. Soaring Dog, you got it. You can still do this. You got one more
We're on
question.
the board at least.
DNA can accidentally be altered or damaged by genetic engineering, in vitro, mitosis, or carcinogens. And the big key word here is altered by accident. All right. So yeah, that was a tough one. So it was being accidentally altered or damaged. That'd be through those carcinogens, which, as I said at the beginning of the Kahoot, they are chemicals and toxins that are that can cause cancer, which is typically where we see the errors in cell division, and can actually alter the DNA there. Yeah, so genetic engineering can alter your DNA, but that is on purpose, not by accident, okay? So that was a tough one. All right, we didn't get like the big grand like animation with our podium. This is sad, so maybe it's only because we had 2 people play. But
Right.
good job, guys.
Kahoot. Yay! There's no t.
Yeah, there's a Kahoot. But yeah, so it's like these daily Kahoots are hard, just because you're learning them that day, or maybe a couple days before, so you don't really get a chance to really much process the information. So honestly, great job, and great job for continuing to come on out to these.